and if the microorganisms you encounter, if they are pathogens, you are going to get sick. And so the question is, if somebody added biofilm, because they are not normal cells, right, somewhat defective cells, and if they were added to food, are you going to get sick? So we asked that question. So we try to study the differences in the pathogenesis between sessile and planktonic cells. So as I said, I'm going to use Listeria as an example, as a model to study that. Okay. The reason we study Listeria, if you are not familiar with this organism, this is a serious foodborne pathogen, and it's not a typical foodborne pathogen, but it causes systemic disease, and you can find them in the environment, animals and humans, and they grow at wide range of temperature. They are resistant to acids and salts, a number of sanitizers or antimicrobials, and they also form biofilm, okay, in number of different environments. So we wanted to study if this organism going to have a differences in their pathogenicity. So I showed you before a little bit that if you add them tunicamycin, you can disrupt their biofilm capacity, but they can naturally form nice biofilm when you grow them in the laboratory conditions. Just a little bit about its pathogenesis, means how does Listeria make you sick. And so the way to study is that it's a foodborne pathogen. And this is a typical Listeria monocytogenes electron microscopy photograph. And since it's a foodborne pathogen, you have to consume it. It's like 100% foodborne. Sometimes other form it can get into your body. And so once it is in the food, you eat it, and then it absorbs through the intestinal tract. And if you are familiar with the anatomy of the human body, so you know that we have lymph nodes called mesenteric lymph nodes in the gut, and that's where the most of the pathogens are absorbed. Then it can get to the blood circulation. You can see them in the liver, spleen, and essentially it can reach to the brain. And also in pregnant women, it causes abortion and fetal death, or premature birth. So because fetus doesn't have immunity, and that's why they are very susceptible. And this disease is very similar to Toxoplasma gondii, which is another parasite that also causes similar type of disease. Okay, and so it doesn't cause much gastroenteritis, but very little. That means you may not see diarrhea, okay, or stomach problem, but it becomes more systemic disease. The other bad news about this organism is that if you had Listeria today, you will not know until like 2 weeks or 3 weeks later, and it's a long term. And in pregnant women, it's even longer, right? Unless somebody is pregnant, had Listeria, and then next thing you know, the fetus is aborted, or the fetus is dead, or if you have a full-term pregnancy and the birth could be infected fetus, there's a question that whether fetus is going to survive. So it's a huge concern for pregnant women, and when there is outbreak, it's huge issue. And I don't know about India; they don't do some kind of surveillance we have in US, but I can guarantee you that this organism is everywhere. Okay, a lot of times the doctors don't know what caused it. What is the reason for somebody lost their fetus or premature birth, right? And a lot of those cases are happening, and I heard those stories. But the bottom line is that this organism causes systemic disease, and abortion and neonatal listeriosis is also very common in pregnant mothers, because our immune system is weak and this organism can infect. So that's the background about this organism as infection. The cattle also get infected, the sheep also get infected, and these animals can also be the source of Listeria. Okay. Now, I got to give you a little bit about its mechanism, how it causes disease. And if you look at this, it produces, if this is the Listeria producing a lot of different types of virulence factors, pathogenic factors, and all of these factors help the Listeria to get inside the cell and then get to brain or other tissues. Okay, those who don't have much microbiology background try to understand the story. Like if you have pathogens, they are getting into the cell, and they are using a lot of these virulence factors. So this is, for example, one of the important pathogenic factor and the receptor, and the other one we've been studying called hits of protein 60, and this is called Listeria adhesion protein. So I mean Listeria has to have a lot of proteins, right, on their surface, and as I mentioned yesterday, a lot of those proteins serve as a biofilm forming protein, okay, and also some of the proteins help infect host, okay, so they have a dual function. So this protein has a dual function, Listeria adhesion protein, and they do all kinds of stuff, so helping the bacteria to go through. So if you take in the laboratory, this is what our laboratory pictures. All this data I am going to show, this is from our laboratory, and this is Listeria electron microscopy. So if you grow them with the intestinal cells, because that's where they go through, and if you grow them, this is called differentiated Echo2 cells. We grow them, and then when LM touches, and you can see it's trying to get inside. His whole job is to get inside the cell, okay, because it's an intracellular pathogen, and then you can see this is already got inside. These are simple studies you can do, but this is how they get in. But more complex picture I am trying to present to you is that, imagine the bacteria is inside the lumen in your gut, right? How do they pass the gut wall? Okay, we got epithelial cells, you got mucus layer, right? And then the bacteria has to pass through the mucus and then get to the epithelial cells, right? And then go through that and get into the blood circulation, because they need to get to liver, they need to go to spleen, they need to get to brain, they need to go to also placenta in pregnant women. So all of those places they need to go. This is their job. This is how they cause disease. So to make a simple statement is that how do they go through the gut barrier? That's an important question, right? Because the gut barrier is very solid. We have epithelial cell lining, and unless you cause damage, it cannot go through. And Listeria doesn't cause diarrhea, I mentioned that. It's not Salmonella, it's not E. coli. It has a totally different function. So one of the mechanisms, we said, well, that's what we want to explore. And this looks very complex, but imagine this is one of the epithelial cells, this is another epithelial cell. So when the bacteria comes in, we found out it binds to one receptor, just to understand the story, called hits of protein 60. And then it initiates a series of cascade of signaling event, or it called, okay, so NF kappa B activation, or it's called signaling cascade. So when the signaling cascade happens, ultimately you get some inflammatory cytokines released, and then other thing it does also, it activates called myosin light chain kinase, and it activates an enzyme. That enzyme, what it does actually, moves the protein which is holding two cells together. Okay, and if you look at the cells, I will use an example. Think about the cloth we are wearing, right? These are all stitched, right, on a thread. Sometimes if you remove that thread, you can remove that, right? You pull one end and pull it. What will happen? Two pieces opens up, right? Imagine that happens in the cells. These two cells were, short like this. Okay, this is called claudin, occludin, ecadherin, all of these proteins holding this cell line together. Okay, so it's like a closed door, right? And how do you open the door? That's the question. Because if you don't open the door, you cannot get in, right? It's pretty simple. Okay, so the way the bacteria does that is that as soon as it goes through this and it activates, it kind of sends a signal, and then these proteins, which are like a thread or like a lock in the door, it gradually opens up. It opens up because bacteria initiated that process. Okay, it's called induction, and that leads to the opening, and then the bacteria can easily go through, right? So this opening of the tight junction barrier is extremely important pathway for many pathogens to enter our system. So if you take Helicobacter pylori, I think somebody mentioned about that. Helicobacter pylori causes gastric cancer, and also they go through pathway same way. Salmonella can also use that, E. coli also, Campylobacter, many pathogens, they have learned a trick how to go through our system. So we figure out that, and so there are a lot of proteins. These are many years of research, and then the bacteria easily pass through. Okay, this is the way to do it. Remember this mechanism, because last day or day before last, I will talk a little bit more about this, and that's the reason I'm spending time. So this is like a simple schematic showing you how if this epithelial layer and how pathogens, any enteric pathogens, which pass through our gut barrier, and they use all of this pathway. So this is, we call it paracellular translocation. It opens up, it goes through it. Okay. There is another way. It's called internally mediated. This is intracellular pathway. And other thing I didn't tell you that if you look at the epithelial cells in the gut, the cells continue to die. Okay, you know that our cells are not permanent, right? How long their life? Anybody knows? How long those cells live in our body? What is the actual lifespan, or how long you think they are surviving in our body? Anybody? What is the average life of cells in our body? So all those epithelial cells, endothelial cells, all kinds of cells we have, right, in our body, we are made of cells. What is the average life of those cells? Come on. Don't feel bad. Just give me a number. How many days, or how many years, or how many months? I mean, average life we live, 70, 80, whatever, maybe. That's not the life of the cells, right? Come on, give me a number. Huh? Say that again. 2 weeks. Okay, that's good number. Anybody else? That's very good. Did you Google it? Okay, that's exactly, yeah, about 12 to 14 days. You know, so she got the right number. So average life of the cells, this is, you should know. If you are studying microbiology or any biology, you should know average life of the cells. Because our cells in the body, they are born and they die. Okay, they are not permanent, except the nerve cells. Nerve cells have a long life. If you have a damage in the nerve, it takes forever to repair. That's why the paralysis patients, they suffer longer. But any other diseases, if you are not cancer cell, if you are normal healthy cell, you have a short duration of life. So that means our stem cells in the body continuously making these cells, right? And then these cells mature, then they die. In the gut, gastrointestinal tract, throughout the GI tract from mouth to all the way to the rectum, the epithelial cells lining, okay, they are born, they die within 12 to 14 days. Why it is important? Because in order to understand any of those pathogenesis and the microbial interaction, this is a very important information. Okay, keep that in mind. One of the pictures right behind me telling you that when these cells die and they are pushed, they leave. Okay, so it's called cell extrusion, and this is continuously happening. So body, what it does also sometimes, if pathogen attaching their cells, they will try to remove that cell from the body. So it's called sloughing. It slough off. The bacteria and the cell goes. So when you are suffering from diarrhea or some dysentery, any of those gastric problems, these epithelial cell lining totally wipes out. They die pretty quickly because microbes kill that. But in natural process, these cells, I think it looks like this, like a tube, right? It's like a mini heel. You know, this is called villi, and those epithelial cells, they are generated in the stem cell below, then move up as they mature, and as they die, they fall off from the tip. And what happens when these cells die? They open up those junction in between, right? And some bacteria, if it is hanging there, they are waiting for that opportunity, so they will go through. So this is what happens. If the cell die, get out, and then Listeria can get in. Don't blame it to Listeria only. A lot of those pathogens, they get through. I'm trying to tell you how they get in, right? This is also another way. We have mucus secreted, and when the mucus is excreted, it creates a hole, right? This big hole, and then the bacteria can go through this also. These are natural process, and And then the other one, you heard about M cell, microfold cell in the gut. And these cells also naturally phagocytic. Their job is to bacteria, some food also can go through those M cells to cross the barrier. Okay. Once they are underneath, they are pretty much free. Now they will encounter immune cells. Immune cells will try to kill them. So I explain you to entire pathogenic model how pathogens enter our body before they make us sick. So this is one way the bacteria can get in. There is another one also called dendritic cell. They also sample bacteria in the lumen and they bring them in. So just to tell you, this is how bacteria or pathogens, when they enter in the gut, and this is how they get into the blood circulation. That give you some idea? Big picture? Okay. All right. So what I was trying to teach you, I know these are some of these models. So we ask that question: is that sessile versus planktonic cell, are they going to have differences in pathogenesis? This is just showing the biofilm formation. The way we assay it, I think you are going to learn today about the crystal violet staining. I think that's the laboratory training you are going to have. And so if you stain these cells on the plastic or glass slide, and with the crystal violet, if they are forming biofilm, you will see this thick layer. Okay, and if you treat it with the alcohol, this dye will come out, and so you can measure the absorbance of the dye. And then if you have a more or higher concentration of dye, that means you have a stronger biofilm. Okay, if you have lower amount of dye, that means weaker biofilm. If you have no, that means no biofilm formation. So this is how you can quantify any organisms they are making biofilm, right? And so let's say I had how many? There are over 100 Listeria isolates. Then we wanted to see how they form the biofilm, if there is a difference in the biofilm forming capacity. I think yesterday you saw some data. So they vary, right? So this strain at the bottom, they are very weak biofilm former. They don't form biofilm much. But the one on the top here, it forms a strong biofilm. Same way these clinical isolates, you can see a wide range of biofilm formation. So to study this, we wanted to see if you take a strong biofilm former versus the weak biofilm former. I'm not going through the details of all this data, but the purpose of understanding the planktonic versus sessile cells, we picked up a couple of organisms from, say, high biofilm former to the intermediate. These are some of those we picked it up to use in our study. You can compare their biofilm formation, and so then we looked at the morphology though. So surprisingly, Listeria from the biofilm, at least this 24-hour biofilm, we didn't see much differences in the morphology. That's kind of good or bad, but this is what we see. Not all organisms going to change their morphology, okay? And so that's what we saw. Planktonic or sessile, they maintain pretty much similar shape and size. So one thing we wanted to see, if you have a planktonic cell, right, fresh, and then sessile cell means we formed the biofilm, and then we scraped off. We didn't allow them to grow. We didn't give them additional nutrients. We just took the cells directly from the biofilm. You can collect them, okay, by scraping, and different way you can remove them. And you take those cells. Make sure sessile cells never convert into planktonic, okay? Then this experiment is going to fail. So you have to be extremely careful. Now you take those cells. Let's see, first step in infection is the attachment, right? They need to attach to the epithelial cell. So we use this called KECO2 cell. I mentioned that before. That's intestinal epithelial cell. And let's look at this biofilm and planktonic. If you look at the adhesion of all of these, can you clearly see the difference? This is planktonic. This is biofilm. Means the sessile cell. So sessile cell, already I mentioned, they are really different types of cells. They are not producing all those adhesion proteins. Look at this. All this 4 stain. This is one of the deadliest pathogens. It's highly infective. This F4244. And you can see this planktonic has a higher adhesion compared to the biofilm forming or sessile cells. So clearly this says they don't attach very well. So instantly told me, oh, looks like these sessile cells are not infective, right? This is what tells me. Well, let's look at it in invasion, because they need to get inside. And same data, look at this. High planktonic cells, highly invasive, Look at the percentage of cells got in, and whereas the biofilm, very little invasion. So we are totally surprised seeing this data. We didn't expect that, but that's what was the hypothesis, that sessile cells are going to be less infected because they are already under stress conditions. They are not expressing all the virulence factors. So that's what we expected. And then another thing, we said that translocation or paracellular route, this is called transwell assay. So you can grow your epithelial cells in a membrane, transwell, and if you add your bacteria on the top, if they are going through the paracellular route in between cells, and you can count them at the bottom layer, and we also looked at that, and you can again see the same result, that planktonic cells has a high level of translocation, whereas the biofilm or sessile cells are very less. So as soon as anybody sees this data saying that, oh, sessile cells are non-pathogenic or not infective as planktonic cells, we are discussing, right, that proved that point. But then the whole idea started thinking, we still get infected, right? Because these cells are pathogens only for certain moment of time in biofilm under stress conditions. They are not expressing those pathogenic factors. That makes sense, right? So we need to prove that. If I stop my experiment right here and you try to publish it, what would be your conclusion? You would say sessile cells are non-pathogenic or avirulent, right? Case closed. And then you said, well, I want to publish my paper. And people are going to believe that, yes, your experiment was good, but that's untrue, right? In bigger picture, in reality. Because we still get sick. They still can cause disease. So we didn't want to stop that, right? So next we wanted to see, well, let's look at that in actual animal systems. Let's see if this case is true. So in order to do that, this is just to show you all those protein expression I mentioned. These proteins are also not expressed in sessile cells. One of them, let's take it, virulence factor, hemolysin. They make hemolysin, okay? And if you look at the planktonic cell, makes a large amount of this hemolysin, and if you look at there in the biofilm, look at this, they didn't make barely any toxin, okay? This is more like a protein level verification. And we talked about this lab protein or internalin A protein. Also in planktonic, you will see high level. They are important virulence factors, right? And biofilm or sessile cells, they barely express. So yesterday or day before, I keep on saying that if microbes coming from biofilm or stressful environment or even saprophytes, right? We mentioned that saprophytic lifestyle from soil or some other unusual environment, they don't need those virulence factors. So that's why they turn it off. They don't make it, right? But as soon as they're in the body, they are going to wake up and they're going to make those to make you sick, okay? And so that's what happens outside. Okay, I will not spend more time. Let's do that mouse study, okay? What we did, let's design. So we have again the biofilm cells or the sessile cells and planktonic cells. You feed them to the animals orally because you get the oral infection, right? Then every 12 hours, this is 12 hours, 24 hours, 48 hours, we sacrifice the animals and look for this bacteria in liver, in spleen and other tissues to see how much they invaded. Make sense? This is how we know how well they are infected. So at 12 hours, I know this is a big table, trying to make you understand a little bit. So this is a intestinal, jejunum, ileum, cecum, colon. Then this one is mesenteric lymph node, liver, spleen and kidney. So what we are looking at, how many bacteria arrive to these organs, right? And since the number is so low after 12 hours of infection, so we wanted to say yes or no. Means we use some assays to say if the Listeria present in these organs or not. So if you look at it, the biofilm almost, look at this, 0 out of 5 mice, we could not detect any Listeria in these organs, except 1 out of 5 in cecum and colon for these, right? But planktonic, you can see a higher number. You see this one? Because planktonic are active, they can spread very quickly within 12 hours. So up to 12 hours, biofilm cells, it looks like they are not doing anything, right? The cells. And this is also these other strains we verified, exactly similar data. But then we are asking, that's only 12 hours. Let's look at 24 hours. Okay, 24 hours, we still saw some difference. Okay, but these differences are getting smaller between sessile and planktonic. Okay, you can still see this if you follow these graphs. Okay, but the most important one, when you looked after 48 hours of infection, the story changed. Do you see this one? All sessile cells become like now planktonic, right? They are in a favorable condition. They converted, they no longer behaving like a non-pathogen. You can clearly see that they are all becoming infective. And most importantly, what happened is that after 48 hours, those sessile cells become more aggressive, more pathogenic than the planktonic cells. Do you believe that? This is kind of like they were sleeping giants, right? They are not infective at the early stage. But as soon as they arrived in the gut, this is gut environment. I think you are doing this, different parts of the gut study. This is in vitro simulated fluid, and we wanted to look at the protein expression and also their behavior. So the storyline is that those organisms started to express, the sessile cells started to become planktonic, and they started to express even at higher level of those proteins when they're in the gut, because we analyzed the gut sample. We took the gut sample from the animals, which are actually coming from the mouse gut, and we are looking for those virulence factors. And if you look at it, LAP and internalin A, especially LAP expression, significantly increased in the sessile cells than the planktonic cells. So what is the storyline? I didn't want to go details of this. What is the storyline? What did you learn? Sessile cells are still pathogenic, right? We all agree. And what they did, because they were in the stress conditions, remember the word stress, right? Whenever some organisms are under stress, they initially look like they are innocent or non-pathogenic or virulent. But eventually they had aggressive behavior than the plankton. So lot of this stress make these bacteria well adjusted or well adapted for the gut, because the gut is a stressful environment, right? We talked about acid, we talked about bile, we talked about antimicrobial agents. We got lot of barriers in the gut, and that's unfavorable, oxygen deficient. I used that word before. And because those stressed organisms, they already adapted themselves, and when they got into the gut, they become more aggressive. And so everything we learned from this actually showing that these organisms are more virulent. So introducing stress to microorganisms, you have to be extremely careful. Try not to introduce stress to these microorganisms, because they're going to come back and going to be more virulent and invasive. And that's the importance of stress. Everybody clear? Any questions on this? I didn't want to go more details. I just want to give you the big picture. If you want to learn more, you can ask me questions later or now. And this, I thought that was kind of fascinating. It was published in Nature Biofilm Microbiome, I think some article. I don't know whether I have cited this or not. Yeah, that one. This is where this whole thing was published. Anyway, so I thought that's kind of fascinating, talking about biofilm cells. They're equally pathogenic. Okay. I think I will end it here. I think that was the last part of the. See if you have any questions. Go to quorum sensing now.
Sir, please tell me then how they can pass blood-brain barrier, listeria?
Oh, how do they pass? Boy,
Yes,
it will take an hour to explain you. No, blood-brain barrier also like a gut barrier.
Okay.
They have a similar structure.
Okay.
And so we believe they have also tight junction proteins. or also similar epithelial structure, and I think we are working on it right now. Other people have also worked on it, and so other people showed that internally in ape, one of the Listeria proteins, help bacteria to go through the cross the blood-brain barrier. But in our studies also we know LAP binds to this HSP60 molecule, and also it can activate signaling cascade or myosin light chain kinase, because those proteins are also involved. And so if it opens up, so it will be like a gut and the brain very similar way. So this may be playing important role. Then other thing is that how do they cross also the placental barrier, because for the fetus. And that mechanism would be slightly different because different types of cells are involved.
Okay. As you mentioned, the M cell is the natural phagocyte.
Yeah, phagocyte.
Yes. Then how can they pass Listeria inside the body? Means it's a natural phagocyte. Then its main work to destroy the Listeria. Then
Yeah. So anytime we have a disease, right? So that means our immune system failed, right? That means immune system could not contain the infection. So the Listeria being intracellular pathogens, when they get into the macrophage and they get out of the vacuole very quickly, and then immediately they start multiplying in the macrophage, and then eventually they kill the macrophage. Okay, if the macrophage is dead, obviously the bacteria is going to survive very well. And so the M cell is a natural phagocytic cell, but the problem with the M cell doesn't kill bacteria. It's kind of like a passageway. Okay, but as soon as they pass, then the phagocytic cells, whether it's a dendritic cells, then the macrophage or neutrophil or other cells, they will try to contain it. But thing is, because Listeria can survive inside, they can kill the immune cells, and that's why they can persist the infection. So anytime your immune cells fail, that's when you become sick, right? Otherwise, if you are not sick or under, little bit of under the weather, that means your immune system trying to work to contain their infection. But if you have a full-blown infection, means your immune system could not do its job. It could not protect you.
thank you, sir.
Okay. Any other questions? Okay.
Good morning, sir. So
Yeah.
my question is, can sessile phenotypes exist without the extracellular matrix?
Say that again. Can you repeat that?
Can sessile phenotypes exist without the extracellular matrix?
Slow down a little bit. Cell phenotype, serotype.
Sessile phenotypes. Can they exist without the ECM, extracellular matrix?
Which bacteria? I mean, you are talking about Listeria?
Yes, sir.
Oh yeah, they can survive without the extracellular matrix. But you are saying that sessile cells, when you are collecting from the biofilm,
Yes, sir.
yeah, there is, when you isolate them, obviously those extracellular matrix are washed off. So that is the question you are asking, or the?
Yes, sir. This one.
Yeah, so that's the difference. Yeah, it's not truly we are injecting biofilm, right? What we did, we scraped off, and it become a cell suspension. Okay, and those the cells we are taking it. But without the EPS, they're still maintaining their, that sessile conditions. You see this? So the sessile conditions would be the genes are down-regulated. They are not producing all those virulence factors or proteins. They're still maintaining for certain duration. But you have to be careful how you keep them. You have to maintain in a water or buffer without giving them any nutrient or food, and so that they maintain their sessile status. Yeah,
Thank
they
you,
will
sir.
survive. Okay. Any other questions? So I will ask you a question. So I think he brought up a good point. So if you have a pure water, sterile water, right? So you have a deionized water we use, right? And sometimes we call it double deionized water, right? No ionic species, nothing. So if you add bacteria, okay, do you expect bacteria to grow in that water sample? It's not a tricky question. Got a pure deionized water, right? Assuming there is no nutrient, nothing, not even ions, right? Deionized, you remove the ions. So if you have a bacteria, freshly grown bacteria you had, say you added, 100 cells into 1 milliliter of water, and I ask you, tell me if this microbe growing in that water. Yes or no? Say that again. So you are saying they are not going to grow. Yeah, so they are not going to grow. That's what you are saying. But these are fresh cells. I took it from a broth. Huh? Okay, so you are saying they are going to. Okay, anybody else? That's one your answer. That's right. So what's your answer? Is it going to grow or no growth? No growth. Anybody else? Huh? He's smart. So some of you are going to say yes or no, right? Overall, you are saying no, right? Okay. So here is the other thing you got to think about. So the bacteria you harvested from a fresh media, they grew, right? So they are healthy cells. Now I took these cells and put it in water, right? Deionized water. There is no nutrient, nothing. So a question I was asking, are they going to grow? Right? Some of you say no, because there is no food, right? We will learn that. That's true. There is no food in that water, right? But this bacteria came from healthy environment, right? They are going to multiply at least until they exhaust their stored energy. So the answer is yes, limited growth. They may grow a little bit, then after that they're going to stop because they have the stored energy. So a lot of times I think students do a mistake. It's a perception that, oh, they are not going to grow. But you are forgetting they can multiply their number. So how you design your experiment, even in the buffer they can multiply, they can misrepresent your number. Keep that in mind. All those little tricky things, they can multiply to an, if you have a 10 to the 6, they can double it, right? 1 times 10 to the 6, they become 2 times 10 to the 6. So that's a huge difference, 1 million to 2 million cells. Okay, everybody got it? Those who are that side, I think you see this what I was talking about? The bacteria they need food, right, to grow. But in this case, they already have their stored food. Like we can skip a meal or 2 meals, 3 meals, we can still survive, right? Because our stored energy. And the bacteria, when they're growing in rich media, they harvest a lot of energy. They store some sugar inside. They can still continue their process at least for a 1 generation or 2 generations. So they will multiply at least 1 time or 2 times depending on how robust they are. Otherwise, if you have a starved cell or sessile cells, you put it in the water, no, I don't expect them to grow. You see this? So it's all physiology. Okay, I thought I will ask that question. It's not tricky question though. So we're going to talk about the quorum sensing. I know this presentation should not be that long, and I know I'm pretty near, but we'll try to stay on time to give you the break. So quorum sensing and the regulation. So from Monday morning all the way to yesterday evening, everybody talked about quorum sensing, right? It's a language, it's a vocabulary of biofilm, right? Anytime you talk about biofilm, you cannot avoid the word QS or quorum sensing, right? It's the cell-to-cell communication. Everybody tells you that. So we'll learn a little bit today, okay? So part of the biofilm formation is that attaches, and attachment is important, right? We all learn that. It's critical. And then it have a quorum sensing, means it communicate each other. And then we need to understand how the biofilm formation regulated, right? And we'll talk about cyclic di-GMP. So attachment we mentioned repeatedly yesterday, a lot of virulence factor, a lot of proteins, molecules, they are produced by the bacteria, and Carli, fimbriae, flagella, proteins, teichoic acid, I already mentioned that, right? And these help the bacteria to attach to the surface. And so here, before I get into quorum sensing, I want to give you a little bit of background how these proteins are made. Okay, one aspect I introduced protein secretory pathways, right? So I want to ask you, or how do these proteins are produced before they are available for attachment. Okay, and again, these are more advanced knowledge you need to understand, and these are important. These are the way, these are the direction research are going. So this is called protein secretion system. So bacteria or any microorganisms or our cells, they synthesize proteins inside, right? Even the Carli or flagella, all of those molecules are synthesized inside, and then they are exported outside the cell. Okay. The question is, how do they get out of the cell? Okay. And there is a special machinery help the bacteria to release these molecules. Okay, or protein secretion system. So, for example, we went through this gram-positive structure, right? So you have a cell wall, right? That's the peptidoglycan structure. Everybody follow this. Then you got the cytoplasmic membrane, and inside, this is where these proteins are made. So the proteins are inside. So if it is a flagellar protein, for example, right, or it's a fimbrial protein, they are made inside. How do they get out? See this protein, see this is called exoprotein. Have you ever given thought how these proteins are getting out of the cell? Okay, and you got to have some mechanism how they get out. Okay, so same way in the gram negative. Gram negative even difficult, right? You got extra layer. So you got outer membrane, then the plasmic space, then this is the peptidoglycan layer or cell wall, right? And then the cytoplasm. So look at these proteins are here. How do these proteins get out? There are 3 layers, right? So this is always an important question. What about the mycobacterium, right? Mycobacterium has again cell wall, but it has this lipid layer outside, extra layer, right? And mycolic acid layer, and then the cytoplasmic layer. So how do this protein get out of this cell? So always a huge question. And then if these proteins are not outside, they cannot use them for pathogenesis. Are you following me? Those are important virulence factor, right? So people study this, how these proteins get out, for the purpose of, if you want to develop a vaccine or if you want to develop a drug, can I block this protein export? So think this number of ways. Everything we are learning try to prevent infection, prevent disease. All your goal would be also, right, I'm trying to study these bacteria or how these proteins are made, because eventually I want to make a vaccine or I want to develop a drug to prevent this protein from getting out. If they stop outside, then they will be less invasive or less infective, or they cannot form biofilm. Are you seeing the picture now? Okay, so that was the reason we want to know this. So I know this picture is a little bit colorful, but complex, right? So we call it protein secretion system, right? I already mentioned that. So there are different types of protein secretion system. So there is a T1 through T9. So type 1 secretion system called T1SS. Okay, and if you look at this in the gram negative, that's where the most complex cell architecture, right? Because you got outer membrane, then you got the cytoplasmic membrane, and then the cell wall in the middle, right? So the question is, how do this protein get out? And look at this, all those proteins. Adhesin, we mentioned about BAP yesterday, right? Few times, bacterial or biofilm associated protein, pili, flagellum, adhesins, all of those, Carli, all of those molecules, how do they get out? So a lot of you, I think I talked to you, you guys said, well, I want to do research, how do I block or I would prevent biofilm formation? Well, I'm going to work on, say, plant extract, for example, right? A lot of times people are looking for bioactive molecules, and if I add that, can it prevent biofilm formation, right? So number of ways you could apply. Look at these drugs or the molecules, can it prevent their secretion of these molecules which are essential for attachment? And if you prevent attachment, of course you are going to prevent their biofilm formation ability. Are you seeing this? This is how you have to connect it. Okay, and so here I want to show you this type 1 through type 9. So this is a type 1. It's almost like called ABC transporter, and it's like a channel. Okay, so proteins are made, it goes through this and get out. Okay, and then display on the surface. Then type 2 also, and that is called sex system, type 2 secretion system, type 3 and type 3b, type 4, N, type 5, type 6, type 7, type 8. So essentially all of them maintain some kind of general structure. It's like a create a tube, right? If you are trying to pass water or air from one room to other room, you use a tube, right? So bacteria, they are using some kind of channel, and that's how they drive their molecules. Okay. So the type 1 and type 2, a lot of this kind of releasing outside. One of the most important one we discussed quite a bit in the disease mechanism is called type 3 secretion system. This is the key, type 3 for many diseases. What it does actually, type 3 secretion system, you heard about the syringe and needle. When you go to see a doctor, what they do? They inject you medicine, right? How do they do that? Use a syringe, right? And so type 3 secretion system is like bacteria use this syringe directly deliver its virulence factor inside the cell. Okay? Directly. The reason is they don't want to get lost. If it is not directly injected, it can get lost. Okay? So I think that's the one. And so if this is flagellar synthesis, and this is the called type 3 secretion system, which directly inject the proteins into the host cell. And I think these are some actually well studied. These are actual electron microscopy photograph of type 3 secretion system in bacteria. These are, I think, could be from Salmonella. A lot of these molecules are produced by Salmonella and directly inject into the epithelial cell. This is how they look like. The cytoplasmic membrane, cell wall and inner structure, outer membrane, and then it injects directly. This is actually model it, you look like this. Okay? So the whole idea being is that gram-negative organism, gram-negative, all the bacteria, notice the Salmonella, E. coli, Escherichia, Campylobacter, these are Vibrio, these are all gram-negative organisms. They have 3 layers and they use this type 3 secretion system to inject a lot of these molecules. But the reason I'm bringing this up for biofilm formation, I already mentioned that these molecules can help release a lot of these molecules outside, okay, for purpose of using them as adhesion factor. I know we need to take a break. It's 11 o'clock. Let's take a break, then we'll come back, then talk about from here, and we'll finish that. Okay? Okay? Take a break, and then during break or after break, you can ask me questions. I'm available. Or if you are here, we'll talk about that. Are you refreshed now? Energized? Okay. All right. So any questions? No, you are shy of asking questions. I know it's not loud. Okay. So we talked about the protein secretion system. This is the gram-negative organism. I think you got general idea, right? I mean, this got this different type of device. That's what you call it, and they use that to physically deliver small molecules from inside to outside, right? Sometimes they could be released directly outside of the cell, like the fimbriae or flagella, and they need that to attach to the surface so that they can form biofilm. And for infection model also, they use these proteins, right? Okay. So these all those 4, the the bottom of these slide sets, there are more descriptive information. So when you receive these slides, make sure if you want to go and read up more, I have it way below there, so you can read it. So this is a structure of, this is flagella. I think I showed you yesterday the flagellar assembly and structures, and it requires so many different proteins to make this flagella. Okay, and when the flagellar proteins are made, a lot of times these proteins can be delivered through this type of machine to get outside. And then once the proteins are outside, they can be assembled or then form the flagella, right? So this just showing an example. And I was telling you type 3 because it's also important for pathogenesis. So directly infecting epithelial cell or during infection, they need to deliver those virulence factors. And so that's what they do. And this is a model, but these are actual electron microscopy photograph of the type 3 protein. In gram positive, we got a simple structure, right? Cell wall, peptidoglycan, and cytoplasmic membrane. And one of the system, we call it SEC system, SEC. SEC come from secretory pathway. And that's the most important one. Even though it got some other molecules, they're showing it, but the SEC system is most important for delivering protein from membrane inside the cell to outside. This is the cell wall, and then it releases outside. And so for gram positive, it's less complex. They don't have a too many system. They have just called SEC system. Okay, so it's easy to remember. But gram negative, I showed you all those, all the way 1 through 9 protein secretion pathway. Okay, all right. So this is called SEC-A2 system. If you are interested to learn how it is done, let's look at it. So this is SEC-A protein. If this is a protein, it has a leader sequence called signal peptide. SEC-A protein binds to it. So this you can see SEC-A protein binding to this protein, and then this goes through called SEC-YEG kind of the channel. Okay, and this is energy dependent. You need ATP energy to actively transport this complex outside. So actually the SEC protein stay behind. Okay, and this protein got out. So this is the protein. This is the leader sequence. Leader sequence or signal peptide doesn't go outside. Okay, but it helps drive the protein. So if you know about protein biochemistry, is that how do they get outside the cell membrane or how outside the cell? Always the protein has to have a leader sequence to get out, right? What about some protein may not have leader sequence? You heard about that? The protein don't have a leader or leader sequence, but those proteins are also getting out of the cell. So they cannot stay inside. They need to be outside. Like I was telling you, lot of proteins bacteria make, which make us sick, or toxin molecules or adhesion factors, they need to be outside the cell in order to have a effect. And so that time, the SEC system is designed in such a way, whether it has a leader sequence or no leader sequence, it can help the protein to be outside. Everybody clear? With or without leader sequence. So that's why the SEC system is important. If you mutate this organism and say SEC-A mutation in SEC-A gene, what will happen to those proteins which are supposed to get out? Are you going to see this protein outside if you make a mutation in this protein? You will not, right? So you are going to make it a defective. So you see this one, what I'm saying, that you can make this bacteria non-pathogenic by manipulating its secretory pathway. I don't have to worry about this virulence factor. Okay, I understand that. But as a microbiologist or those who are thinking of biotechnology or any research, these are the strategy we use Why secretory pathway is important? Because you need to understand how these proteins are getting out of the cells, what mechanism do they use. If you understand the mechanism, can you turn off that mechanism? And we created a mutant of SEC-A2. This is SEC-A2 pathway, right? And we found out that bacteria become actually non-pathogenic. This is the SEC-A2 pathway. This is SEC-A, it's extremely important for SEC-A2. And then we found out that this bacteria become less pathogenic. Think about it. So can you design something to make it ineffective? So these are the strategies you can take it. So not just only one mechanism you can use to inactivate microbes. That's my point. Everybody is talking about antibiotics. Geez, I mean, you need to find an alternative to antibiotics. Let's put it this way. Because we know bacteria become antibiotic resistant, right? They have a nice, elegant tool to become resistant. But then don't give up. You need to understand every other mechanism they use to become pathogenic. Right? Can we alter those mechanistic pathway, and that can also make the organism ineffective? Okay, that's the message I have to bigger message. Because I think I know you guys heard all the time antibiotic resistance, everybody is tired of that, right? But your solution for that problem, you are approaching only limited way, right? But think about all those other ways they make us sick. Okay, let's now spend time of the quorum sensing. Okay, I already defined that before, right? What is quorum? Quorum definition. Again, this is in a written way, so you can follow it, right? The minimum number of members of an assembly or society that must be present at any of its meetings to make the proceedings of that meeting valid. Naturally, if you call a meeting, you have board members or members, right? We vote, we pass the policy, and that's what we call quorum, right? And we need a minimum number, right? So minimum number, then you might ask, what does that minimum number mean, right? So if you have 9 people in the committee, right, what would be your minimum number? So I would say 50%. I would say 51%. That would be minimum number, right? More than half, right? And so 51%, we are not saying 50%, then it's going to be half, right? And so usually when you make this type of board, I don't know whether you know that or not, usually we make even odd numbers, right? So that if you have few people don't come, right, so that when we vote, make sure never tie, right? That's also another requirement. But nonetheless, you need to have a minimum number of people to vote or assemble, right? So when you talk about quorum sensing for bacteria, we actually talk about the numbers. So it's a cell density. The number came from number of bacterial cells, all population, right? So the number means that if I reach a certain number, our system cannot sustain this. Means I don't have enough food to feed all the people or all the bacteria. So that's why they said, well, it reaches certain numbers, then they start to communicate. When they are low number, they are not going to communicate. Everybody clear? And so that's where the quorum sensing comes. So quorum sensing, already I talked to you about. Quorum sensing from Wikipedia, if you read it, quorum sensing or quorum signaling is the process of cell-to-cell communication. Cell want to tell them, other cells, that this is where we are, allows bacteria to detect and respond to cell population, the number. Everybody clear? It's always about the number. Okay? Always about the cell density. And so that's where the main definition of quorum sensing. And stress, I think I talked to you about stress. I'm talking about stress many times. We all suffer from stress. And stress could be another reason why bacteria go and form biofilm, right? Or biofilm also can help make cells stressed, right? And so stress is always so important. Everything I mentioned this morning, all about stress, right? And so stress means any deleterious factor or conditions that adversely affects microbial growth or survival. And I needed to bring that up again so that I can talk a little bit more about it. So microbial stress response, microbes may respond to stress by many ways. How do you respond to stress? You have different way of handling stress, right? Somebody can say, well, I'm so stressed, I would rather go outside and run for a while, or I play badminton, or play or listen music, right? All kinds of different way we want to unwind, right? And that's how to relieve stress, right? But when the stress comes, you respond differently also, right? That's a stress management, what I just said. But somebody may suffer from anxiety, right? Somebody may show high fever, or somebody may be sitting one corner. This is how an individually we respond to stress. If you think about microbes, microbes are exactly like us. They will respond differently. Not all going to respond same way. Okay? So that's what I was trying to say is that stress response, what they do as soon as they arrive into the stress, they will produce some specific proteins that will help them to cope stress. Okay? Let's talk about, say, freezing, low temperature, right? Cold condition. Microbes, some microbes like cold and some don't, right? So those who don't like cold, if you put them at low temperature, they want to still survive, right? They don't want to die. What they're going to do? How do they survive in the freezing condition? So you are going to make certain proteins, right? Usually your membrane, which can be frozen, right? So you alter certain proteins or the structure of the membrane so that freeze cannot kill you, so that your enzymatic process inside the cell still remain active. Everybody clear? Same way if you are subjected to like 45 degree here continuously, right? You still need to survive at high temperature, right? So how do I do it? So I may produce certain protein which can help me protect my critical function of the brain or heart, right? So I don't die. So microbes also do the same way. Okay? So this is always happen. It's stress adaptation, and they will synthesize or upregulate certain gene, they will downregulate certain gene. I showed you some of those pictures. Certain conditions they will not make any protein. Some conditions they will make some other protein, which is going to help them survive. Okay? So they will make some of those to repair damage, maintain cell function, eliminate the stress agent. That's their purpose, and they are very good at it. And remember, all microorganisms you will be facing all the time, they are all stressed. They are never normal, except the one you grow in the laboratory conditions, right? You provide them the ideal environment, so they are not stressed. So keep that in mind. Transient increases in resistance or tolerance to stressful conditions. And other thing happens is that, I think you haven't thought about it. So if an organism become heat stressed, right? So or say called thermal stress, right? So for example, you cook some food, right? And you had pathogens in that food. You cooked it, but then the organism somehow managed to survive. If it survived in that heat stressed conditions, so that means it has developed some proteins, right? So now it's kind of like a resistant to heat. Everybody go with me so far? But the problem with that same bacteria which become heat tolerant or can withstand heat stress can become resistant to antibiotics. Have you ever thought about it? Remember, always I know all of you maybe have a high concern about antibiotic resistance, right? Then you might be thinking, well, these are the set mechanisms, how they become resistant, right? But have you ever thought that if this organism become cold resistant or heat resistant or acid resistant, can that organism become antibiotic resistant? What about, let's ask this as a question. What do you think? You said yes? How many said yes? Or how many said no? Raise your hand. So some of you said yes, they are going to be antibiotic resistant. Some of you did not raise hand. You are unsure, or your answer is no, or unsure, which is the correct answer or correct response? Unsure, right? Yeah, I mean, I would be unsure too. Well, you listen to me today, right? From now on, think about it. Stress, I'm going to talk towards the end of this lecture. That's the problem. We are not paying or thinking in a dimension would be able to address it. When an organism become resistant to certain stress, oftentimes they display certain behavior where they become resistant to other stress also. And especially you need to start thinking that heat stress organism become resistant to antibiotics or antimicrobial. You couldn't connect it yet. The word I used is called cross protection. See this one? There is a reason why I use this terminology called cross protection. Cross protection is that you are protecting yourself from one factor, but in the meantime, you made yourself such a way that you can also defend another threat. That's what I meant. Right? So think about you are doing physical activity, right? You built up your muscle and you said, okay, or you learned karate, right? Think about it, right? And so if some bad person is coming, one person, you can defend, right? Then there might be another person who may not be karate specialist, but he has another way of, attacking you. Can you protect against that another threat? See this one, that I was thinking. So you prepared in a way. A lot of times, I can have a multi-defense strategy, and microbes has that. And oftentimes we don't think about it. You have to understand, a lot of times this is called master regulatory, somebody controlling on the top. That regulatory element, if it gets activated, everything underneath that, they are all going to be activated. Okay, I'll come back and the end I will explain you more. Don't get confused. Okay, dormant state, sporulation, formation, VBNC, we already mentioned that. If they are under stress, they will find this pathway to protect themselves, right? If I convert into VBNC, I'm protected for a while, right? I'm not dead, but I'm protected, like a persister, right? Sporulation or force, adaptation, starvation and oxidative stress also can induce biofilm formation and promote persister trait. Already mentioned that. Everybody with me so far? Okay. Stress-induced virulence factors promote biofilm formation. Again, repeating same theme. These are the proteins. Due to stress, they can overproduce it. They can help them initiate, and then they become more adapted to the stressful condition. So now that brings us to cell-to-cell communication. How do the cells communicate? So as I mentioned, or many people mentioned already, they make small molecules, right? They secrete those small molecules. And in general, those small molecules, there are 3 groups right now. I'll give you 4. Acyl homoserine lactone, AHL, autoinducer, AI, autoinducer protein, autoinducer 2. This is called autoinducer 1, also AHL. So when this autoinducer come contact with the cell, it binds to the receptor, and then it activates set of genes which can lead the bacteria to form biofilm. Or might say, well, I don't need to do biofilm. Can I just move? Motility. Can I increase our pathogenesis? Can I produce some pigment or luminescence? A lot of those factors. So, but these molecules are produced by this bacteria, right? So it synthesizes this autoinducer, then can come back inside the same cell or nearby cells. Everybody clear? So they are making small molecules as a communication system. They are sending signal, and that signal eventually helping the microbes to cope with the environment, what it is. Okay, so this quorum sensing starts with stress condition, with the nutrient starvation, low water activity, high temperature, antimicrobial compounds, acids, other microbes, and they will produce this auto-inducing molecules. And these molecules are going to bind to a receptor, is going to express certain genes, and as a result, that genes going to help to make the organism adapted to that environment, whether it's a biofilm formation, right, sporulation or whatever may be. Everybody clear on this? Okay. So there are 4 types of molecules. One is called autoinducer 1. That's the one I mentioned. It's called AHL. They also called N-acyl homoserine lactone, and it's produced by gram-negative bacteria, and it's called intraspecies communication. So the gram-negative can communicate with the gram-negative bacteria, okay, within this community. So this is the AHL, and so well-studied molecule. And so, for example, this picture, everyone can read this, even on those people who are in the back. Okay. So, for example, AHL, N-acyl homoserine lactone, is actually encoded by Lux cassette or Lux gene, and when it is activated, so it's going to make this molecule. Then this molecule is going to come back and going to bind to the receptor, and then it's going to transcribe the gene, and that gene, again I'm repeating, that can help the bacteria to be part of the biofilm. It can help the bacteria if it is a spore former, it forms spores. Also, it can make it very pathogenic, right? Produce more virulence factor. It can also increase the motility, and it can also increase bioluminescence. So this is in Vibrio that works very well. And that molecule, when it is secreted, is essentially sending the signal to the cells that this bacteria then is a cell density dependent, means population dependent. Okay, at certain level, they are going to make this. So this is how it works, right? Then there is another one called autoinducer 2. It's produced by both gram-positive and gram-negative bacteria, and it's called universal signaling molecule. Okay. Then the autoinducer 3 usually is one. This is enterohemorrhagic E. coli, the one which causes kidney damage or kidney failure or bloody diarrhea. They have identified that this is present in this organism. Autoinducing peptides are produced by gram-positive bacteria. So this is exclusively mostly in the gram-positive, this is exclusively in the gram-negative, and AI2 is both gram-positive and gram-negative. Everybody clear? And again, they are going to make it in the environment where they are in. So people who are working in foodborne pathogens, they have isolated these molecules in the milk, meat, vegetables, various food products. So what does it mean? If the bacteria grow in any environment, they are going to produce it, right? And so that's a good indication they are making this molecule, right? They are communicating, cell-to-cell communication. Later on, we will say that how can you control these microbes or prevent their growth by using molecules that block this communication. Have you thought about it? That's the reason we study again. They communicate by using these small molecules. And now, can we use to block the production of this molecule or neutralize this molecule? Then they cannot communicate, right? Then they will overgrow, right? Keep on growing, there is no nutrient. Eventually, they are all going to die, or they are not going to be less. They are going to be less pathogenic, less virulent. Okay, so those are the strategy. We will do that later on. Let's look at it. So as I said, this is concentration dependent. So at low concentration, you are not going to see much production, but at high concentration, when the cell population increases certain number, you will see these molecules are produced, and that's when it works. So in the bacterial growth curve, okay, let's talk about the growth curve, right? You see the growth, right? And then after log phase growth, they kind of plateau, right? And then eventually they slow down, right? So if you look at the growth curve, where you are going to see more this molecule production, stationary phase, right? Because that's where it reaches the maximum cell density, and that stage, because there is no nutrient, right? That's they stop growing. They exhausted all the nutrient. They reach to a certain level, and that's why they're going to make a lot more molecules. So if you think that scenario in a biofilm, right? So they reach certain cell mass, and there is a lack of nutrient, and that's when they start making more molecule. And that way they are going to ask the community, said, stop multiplying, stop dividing, because you don't have any more food. That's how they use it. So this is the cell-to-cell communication, helping cells become part of the biofilm formation. In gram-positive bacteria, this is in, you can say gram-positive or gram-negative. In gram-positive, so the auto-inducing peptide I mentioned, look at this, it's synthesized, it's secreted outside the ABC transporter, and it binds to this receptor, histidine kinase, then it comes inside, it binds to the gene during transcription, right? All of you know how the protein is made in cell? You should, right? The central dogma, right? DNA, RNA, then protein, right? And in order to make the protein, so you have to transcribe, right? And so they need to bind to the promoter. So always you can see this transcription happens, right? And then they're making all of those, you already seen this picture, and helping the bacteria in that environment. Okay. Now I'm going to, so those are the small molecules help the bacteria communicate, right? So now I'm going to introduce another small molecule, and which is called cyclic dinucleotide signaling pathway. And that molecule has an important role. So now you start with the GTP, and then GTP, diguanylate cyclase, convert it into called cyclic di-GMP, this molecule. Okay? And this molecule play an important role. Look at this, production of biofilm formation. So as soon as this molecule is formed, it play a key role in biofilm production or biofilm synthesis. So you can see this biofilm, but what it does also at the same time, if this organism happen to be a flagellated organism, I don't need flagella anymore, right? So it turn off. So this it blocks motility. And that's what I was telling you again, repeated yesterday, that during biofilm formation, until it attaches, where it guides the bacteria to get to the site, it attaches and then initiate the biofilm formation, then there is no need for flagellar synthesis, and by that time already this cyclic GMP is made, it stops the motility. Motility is no use. Okay? They will stop flagellar synthesis. So this is how it plays a role, and then it makes 2 GMP. So this is one of the critical signaling molecule in biofilm synthesis. So again, it's like a different way I'm presenting. Again, 2 GTP from this cyclic GMP, and it makes these other small molecules. It has multiple varieties of function in cell, and then also it can interact with the DNA. It's the signaling cascade, how they, if you study more, you'll see that the series of events taking place. It's a cascade, means signaling cascade, one reaction after each other. Okay? So broadly, if you look at it, this is again same information, and cyclic GMP is made, and the biofilm formation. Then during this, it can summarize it. It makes these organisms antibiotic resistant. Okay, besides other function, but the just cyclic GMP can make these organisms resistant to antibiotics. It can increase the virulence factor in this molecule, and also can help evade immune system. Okay, this kind of like a summary of it. I don't want to go detailed because this is really a little bit more complex, but again, some of you can follow a little bit. The cyclic GMP can regulate a lot of those genes during this biofilm synthesis process. This is in Pseudomonas, and how you start, a lot of those genes listed here. And if you want to study a little bit more, in this article, they have clearly described, at least for Pseudomonas aeruginosa, in this pathway, and this role in this whole, different stages of biofilm synthesis. Again, this could be high level for some of you didn't have the background, But here the take home message would be the cyclic di-GMP play an important role in the biofilm synthesis. Okay. I had many slides on this. I just didn't want to talk about details of this, some of those two component signaling system regulated by cyclic di-GMP. Okay, and yeast and mold also have a similar strategy but slightly different from bacteria. They also use these quorum sensing molecules, and only thing is that they also use cyclic AMP and mitogen activated protein kinase pathways for activating some of those signaling cascades. Okay, it's slightly different, but it's a very similar mechanism. Fungi and yeast biofilms are often more recalcitrant to antifungals, like antibiotics. Bacteria don't respond. Antifungal agent also, they don't work very well with the biofilm produced by yeast and mold, and also they have efflux pump, also can spitting out antibiotics. In fermented food systems, bacterial and yeast biofilms can contribute positively by stabilizing microbial communities, producing flavor compounds, preventing pathogen colonization. Anyway, these are the yeast and mold biofilms. Okay. You got some general idea, right? I mean, we talked about quorum sensing molecules and then cyclic GMP, how that regulates the behavior in the biofilm, right? You guys are clear on that side? So you guys are talking a little bit, discussing, right? Okay. So you got the idea, right? Now let's talk about genetic regulation. Minute ago I told you that there is a master regulator, somebody sitting on top, right? That's regulating a lot of genes underneath, right? That's the part I wanted to talk to you. Let's see if you have seen this. Regulation of virulence genes. So during transcription, instructions stored in the DNA are transferred to the messenger RNA. Central dogma, right? Messenger RNA convert into protein. So during this transcription stage, remember again, this is your biochemistry. All of you learned that, I'm sure. That transcription has 3 steps. One is called initiation, elongation, and termination of RNA synthesis, right? That's the mechanism of transcription. And there is RNA polymerase, right? RNA polymerase play an important role in the protein transcription, right? RNA polymerase. And RNA polymerase works best if you have another factor, and we call that called sigma factor, okay, or alternate sigma factor. That sigma factor is the key because it binds to the RNA polymerase and the transcription enhances significantly. Okay, so who is the most important in this case? The sigma factor, right? And all microorganisms have this sigma factor. By any chance you upregulate the sigma factor, or sigma factor is the Yoda. You heard about the Star Wars, right? The little guy, he has the power, right? And to me, I think the Yoda is the sigma factor. Okay, he has all the power, and he can turn the cells into something totally different. You see this? And that sigma factor play an important role in this. So if an organism, right, a heat stress, the sigma factor get upregulated. Okay. Now think about it. Sigma factor got activated and is binding more to the RNA polymerase, and any gene sitting underneath, if I have a drawing board I could have shown you, these are genes underneath all of this, right, in an operon. Everybody knows that. In operon there are a series of genes. Some of them are maybe growth factor, some of them could be virulence factor, all of those genes. And if this regulator get activated, all those genes downstream, they are all going to be activated. Not only the heat stress genes, also the antibiotic resistance, all those other factors are going to be activated. If you see this, what I am saying? So the activation comes from the leader, top. It's like a hierarchy system, right? You have a president or vice chancellor, all below this, right? and we follow his order, right? And he controls everything, and so sigma factor is that guy, that person on the top. He got so much influence what we do underneath. And in the microbial world, that's very important. The thing is, it cannot discriminate stress, heat from cold, because that guy already got activated. And even the antibiotic resistance, also sigma factor can get activated. So if I show you the diagram, I don't know, I didn't show you this, and you can see this. This is the RNA polymerase, right? And this is the sigma factor. It binds, it forms this complex, and as soon as it forms this complex, all those genes located in that, they are all going to be activated. It's the promoter function, right? You know that promoter binds and every gene get activated after that. So in the regulation, in the biofilm, or all those stress factors we mentioned, essentially we are getting upregulation of all those genes downstream. And that's what helped the bacteria survive better. Okay? And that's the primary message. You can read it more. They got a lot of housekeeping genes, means you need housekeeping genes. They get also upregulated. And alternate sigma factor help them bacterial survival, stressful environments, and regulate virulent genes. So it got really a lot of function. People never thought about this, that heat stress can make the organism resistant to antibiotics. So you may be preparing some way to control the pathogens by heating or providing antimicrobial agents, but if you cannot kill it, but that becomes so activated or stressed, now that organism become resistant to antibiotics. And that's the factor nobody's thinking about it. You need to think about just not only traditional resistant mechanism, but it could be I'm doing something to activate this regulatory element and they become more resistant. Okay? So I hope you learned something totally new or you haven't heard about it, but at least this cross protection mechanism we have to pay attention to. The cross protection. It's not just single mechanism. These are the mechanisms actually responsible for making this antibiotic issue the problem, besides all those other factors. Okay? Alright, so you can see this is, I think, Lyme disease. I picked up, they have a different sigma name or number, and you can see this, this sigma factors in Lyme disease, you know that, like Borrelia. This comes from deer, right? It's a debilitating disease. It's very, you'll find it in North America, a lot of cases. And this is in, this also you'll find it in Salmonella, E. coli, a lot of pathogens we have discovered that. And another story is that you are familiar with the International Space Center, ISS, and that's always circulating, right? And that's still functional, and multi-countries, they go there and they live there. And so I was visiting. One time I was in NASA, our space administration office, and you know that NASA, we're trying. I think we're trying to go to Mars, right? I mean, they've gone to Moon is no problem, but the going to Mars is a long many years process, right? At least going there, coming back, they are estimating it will take about 3 years, 3 to 5 years. But one of the issue with the space food, if you have a pathogen in the space, okay, especially in the shuttle, right, when the astronauts traveling, and they also worry about foodborne pathogens, especially if you have food you are consuming, if you have a diarrheal pathogen, Salmonella or E. coli or even Listeria or any of those Campylobacter. You don't want any of those pathogens in that environment, right? And so the astronauts, they worry about foodborne pathogens. They eat food, we send them, and that's a constant problem. And so they always prepared in a manner, it's almost like a sterile food. I don't know if you have seen that or not. In my textbook, there's a picture of freeze-dried egg, what's it called, scrambled egg. You know, have you seen scrambled egg? It's like it's not omelette. Once you make it omelette, and make it into small pieces, and it's called scrambled, right? And then you put it in a pouch, and then you freeze dry it. And when they want to eat it, they can add moisture or water, or also soup, all kinds of meal, they make it freeze dried. And the way they prepare it, there is no problem with the pathogen. So to make the long story short, is that they worry about, so if there is salmonella in those foods, right? And so the astronauts, they're going to get sick, and if somebody has a diarrhea, it's a bad thing in the closed compartment, right? Everything is floating. So the fecal material would be also, you can imagine the picture, right? And that's nasty, and nobody wants to imagine it. Especially the astronaut talking to us. It was kind of like a prestigious panel. I was invited to give a talk or discuss with them. And so they were talking about, well, they are studying salmonella in space. The salmonella in ground, in the Earth atmosphere versus in the microgravity or no gravity, how they behave. Are you getting the picture? Are they going to be equally pathogenic in microgravity environment, the space environment, or in the Earth environment? Okay. I don't know the answer yet, but just to continue this story, the person who is studying this, I invited him and we had really good interaction. He's a space microbiologist. Nature actually publishes a journal called Space Microbiology. I think I forgot the name of it. NPJ Space Microbiology, something like that. You can check it out. And the editor-in-chief or associate editor, actually, this person, who I'm talking to him. And he studied salmonella in space, and he said, you know what, this salmonella, the virulence factor in space is slightly different from the one grows in the Earth. I said, how so? And he was saying actually the central regulatory element. That's what is different. I said, what is that? And so the answer is the RPOS. I think there are different regulatory elements, which is the alternate sigma factor, which is dominant when they're growing in the Earth. If you are infected here, that is the regulatory element, make the salmonella more pathogenic. Okay? But in space, it's actually HFQ. And this is because I saw the HFQ. That's the sigma factor is highly active when it is in the microgravity environment. You see this one? But nonetheless, it also controls this virulent gene. So in order to pathogenicity wise, yes, they are going to be equally pathogenic or more or less, but they have a different regulatory element because it needs more research, right? You have to do animal research in the microgravity to see if the activation of HFQ is going to make that organism more virulent for the people who are in the microgravity environment. You see this one? But at least it's telling us that the regulatory elements were working differently in different gravitational force. Got it? So they could be controlling a lot of other factors. So that was actually the fascinating knowledge I learned from that. And so the central regulatory element going to play an important role in pathogenesis. Got it? Any questions about the quorum sensing and the regulation? Go ahead. Use the microphone. Or you have to speak loud.
So the question is that in clinical setup, if the quorum sensing is responsible for pathogenesis, so can we target that quorum sensing or the regulatory mechanism to tackle the biofilm in the pathogen sector or causing harm to humanity and mankind?
Absolutely yes. I think we'll talk about that controlling and preventing biofilm. Controlling the signaling molecules is one of the latest mechanisms people are trying to do that. So if you know there is a signaling molecule secreted in the food and people are trying to understand that, and can you find some molecules that can prevent that communication between microorganisms? So a lot of those plant-based natural molecules, actually they inhibit this quorum sensing molecule synthesis.
And sir, can these methods also tackle the problem for antimicrobial resistance as an alternative for antibiotics?
Yes, yeah, our answer is yes. I think they could have different pathways to help that. Yeah,
thank you, sir.
Any other question? Go ahead.
Sir, actually, can you explain about the sigma factor one more time? Sigma factor, the regulation of virulence genes.
Oh, can I explain you again?
Yes.
Sigma factor, what I was telling you is that So in the central dogma, let's look at it here. So for a gene transcription, okay, so the RNA polymerase has to bind to the promoter area for the transcription of the messenger RNA, that eventually that's going to convert into protein, right? And what this sigma factor does is that once it binds to this RNA polymerase, it makes it even more active. It makes it more powerful. Let's put it this way. So that binding allow it to transcription of more gene downstream. Okay, so it's the binding. Sigma factor binds to the RNA polymerase, and so that you have a gene transcription is much higher.
What is the purpose of this binding once again? Purpose of the binding of sigma?
Oh, binding is to, it stabilizes the structure.
Okay, thank you.
And once it stabilizes, then transcription really enhances. Okay?
Thank you, sir.
Anything else? So we're going to talk about the next, let's get started. How we're doing. I know it's getting pretty close, right? Let's go to the next talk. Again, read it up. I think there are a lot of conceptually, a lot of detailed materials. So I'm going to give you a general picture, and at the same time, I think you can get an idea what actually the big picture is. Stand up for a while, just for a minute or two. Just stand up. All of you, stand up. The reason you are standing up is that you are sitting there for a while. I know after taking the coffee break, right? And it helps prepare your body circulation, increases. So it increases your attention too, right? Now you can sit down. You know, you need to break the monotone, we call it. You had a question? Go ahead. Cross protection? Okay, that's a good question. We discussed about cross protection. So means, what it means that I was using heat, right? So if the temperature rises like here, and so I have adapted to adjust, to adapt myself to this high heat, so that I produce some proteins or I have some protective device, right? So that I don't feel hot, I don't kill myself, right? So if I have adapted to heat, now I can protect myself against, say for example, freezing cold, okay? Because I was adapted to that one condition, now my body is prepared myself to defend against another adverse condition. That's what is called cross protection. So the example I was using is that if you are resistant, the microorganisms, if they are resistant to heat or heat adapted organism, now they become resistant to antibiotics. Okay, and that's what is cross protection. So the antibiotic may not kill that organism. Well, they could make a heat shock protein as a strategy, right? Heat shock protein is a strategy to heat. Yes, they will make, that's the defensive strategy. I didn't tell you, yes, that could be. Bacteria make a lot of different heat shock proteins, okay? And one of the heat shock proteins I mentioned, but that's the mammalian system. We also make a lot of heat shock proteins, okay? And we study heat shock protein 60 in our research, and which is happen to be a very good biomolecule for, biomarker for also cancer. And that's a lot of interesting story. Yeah, you got the point. Yeah, if you are heat stress, bacteria actually make a lot of heat stress proteins. There are some well known called GROEL. You may have heard about that. That's one of the major heat shock proteins. And those type of proteins really help the bacteria. Same way the acid stress, cold stress, all kinds of series of proteins they make to protect themselves. Okay, you can study up a lot of these details. Okay, hopefully this will be really fun lecture. Again, if you haven't done that, so all of our knowledge about pathogenesis come from human. We don't use human. Can I use it as a model? You are human, right? Can we use you to study bacterial pathogenesis? Can you be a guinea pig? We call that, right? Nobody wants to be. So that's why this is very important, right? We want to use animals or animal-derived cells or organs or tissues to study pathogenesis. So all our knowledge, everything I was sharing, lot come from animals, right? I showed you data this morning, our study, my study, using mouse model. And so this is an important tool for biomedical research. Whether you are working in environmental science, environmental microbiology, medical microbiology, food microbiology, all different disciplines, and you need a lot of these models. So primarily for microbial pathogenesis. So my goal is to explain you all these different models, animals, organs, cell culture, and cell-based sensors. I'm not going to talk today, but that will be more towards the end. Next week, I'll mention a little bit about cell-based sensors. And these models I'm going to use, it's not just only for biofilm or any pathogens, okay, or any microbial organisms or anything you want to test. Even a toxic molecule, doesn't have to be a microbe. Or if you have developed or derived any food grade materials to market or to sell, or if you have a cosmetics you want to produce and you want to see if it's a toxic or non-toxic, you have to use this type of model. So irrespective of any field you are using, if it's related to health, you can use this model. Okay, let's examine what are those we are talking about. Why use animal and cell culture models? Like I said, none of you are going to volunteer. Okay, even a student, they don't want to. But I will tell you, sometimes we have used students or prisoners or some other people, even though voluntarily they will allow. So human volunteers, sometimes they use it, especially in prison system, right? Or think about the COVID. When COVID happened, a lot of times we had to become actually the model to test our vaccine efficacy, right? Then you take the data, then you improve it. Make sure we don't get killed by having this vaccine. So that's why the animals are alternate, okay? And then sometimes animal use also unethical. Can we use cell instead of the whole animal, okay? Or their organs or their tissues. So if I give you a little bit of history as perspective of where and how you started, go back and think about the mine. You heard about the mine, coal mine, for example. Coal mine, when they're mining, they go for mining, they'll go inside underground, right? And the problem with the underground, what happens? What is going to be accumulating in that? People may die. What type of gas builds up? Carbon monoxide, right? Excellent. Carbon monoxide is a huge problem, right? It can happen on a number of situations. Even the car exhaust releases a lot of carbon monoxide. It's a silent killer, right? Doesn't have a smell, nothing. So what they used to do, actually, nobody wants to get killed when they are going for mining. So think about, this is in Wales, part of the UK, right? And in this mining industry, say they take a bird. See this? Can you see this bird? Little bird called canary. This is the bird, and this bird is highly sensitive to carbon monoxide. Okay, so if you take that bird with a mask, and if this bird die, then you know there is a high level of carbon monoxide. If the bird is healthy, moving around, then there is no carbon monoxide. Then you can go in and do the mining, right? And so what does it say? So this bird help detect toxin, right? That's why you call it, bird is being used to help detect or determine if there is a possibility of carbon monoxide in that system. Is it safe to mine, right? And so animals being used. So this is the historical perspective of, we've been using this for many years. Now we use even more, right? We can use mice, chimpanzees, or human, and if you have a pathogens, we look for mortality or morbidity, right? And instead of using the whole animal, can you use the cells? For example, kidney cell or B cell, B lymphocyte. And then if you inject or expose to pathogens, if these cells die, you can easily recognize it, right? And then if there is a whole formation, then you can interrogate it. And so that's why we end up using cells. Now let's give you overall view of the different types of animals being used. What are those animals? So non-human primates, we call it monkey, baboon, or chimpanzees, right? And these are expensive. Actually, higher primates is very expensive. And these are gerbils. You heard about gerbils? These are called desert rat. Okay. Very nice to work with. We used quite a bit. But these are called USDA protected animals. They don't breed very well, but these are very easy to handle or use, and they're very docile. So many people use them as a pet, like hamster or guinea pig, right? So these animals are used. And then, of course, guinea pig, hamsters, rabbits, rats, mice, these are more common. Less common, parrots and gerbils. What about other animals? Fish. Can you use zebrafish? A lot of people are using zebrafish now. It's a good model. Pigs or piglets, cats, dogs and cats, they are also being used. What about alternate to animals? Can we use insects? So this is again becoming extremely popular. The flies, fruit fly, for example, the nuisance big one, get into house and whoosh, make sound, go around, and these are being used, fruit flies, for a number of pathogens. And this also larvae, this is the moth larvae, they are also being used. This is also another one, silk moth larvae, they are also used. And then the last one is the C. elegans, is the parasite, the round one, also being used. And these animals or the larvae, they are highly sensitive to a lot of diseases. And that's the beauty of it. I mean, less expensive, and there are companies that sell this. And you can see this bacterial pathogen, fungal pathogen, people have used it. So what you do, you inject it or orally feed it with the feed, and then you wait to see if they're dying. And also you can study their immune response. Okay, even though their immune system is slightly different than the humans, but you can study a lot of those aspects using this larvae model. And so sometimes it's easier to adopt this type of model than the animal. Okay, because your restriction or ethical issue would be much less stringent. You can put them in the laboratory in a small setup. You can provide them feed. They will grow and multiply, and you can do a lot of experiments. And these are becoming extremely popular nowadays. Okay, so this gives you the data equivalent to animal model. Okay, and so these are very important. A lot of times people are using this round one also, and the petri dish, they grow, and you can use them quite big. So few things you have to consider when you are using them. So you are studying human pathogen in these animals. Make sure the pathogen respond, or these animals respond to these pathogens, right? You need to see the similar symptoms, similar tissue distribution, similar mechanism, because that's very important, right? And if you see that, and that way you will know that you are getting the disease reproduced very well in the model. So that's how you pick the model, right? And so, as I said before, these are the strategy you have to use. And other issue is that we use quite a bit of mice, right? And mice, the reason we use it, because many of them are inbred. You heard about the terminology inbred? And inbred mice, they are very similar genetic makeup, right? So therefore, there is a less difference in your data, right? The study you are going to do, the variation would be much less. And so that's one of the advantages. And because they are small, you can keep them in a smaller space, a lot of them, and they are less expensive to house. And if you are comparing the larger animals, of course, these are easy to use. And I showed you already data with the mice, right? So there are different types of mice breeding. AJ mice, the white mice called black C, black mice, C57BL6. This mice, the black one, is extremely aggressive. I mean, if you have worked with mice, I mean, I never got bitten in my 30 years or 35 years, even more than handling mice. I always do mice experiment. Even day before I left, we are doing a large animal experiment. And one time I got bitten by this because it got so mean and mad. Usually these bulb Cs are very docile, and this AJ are also very docile, very easy to handle. This is called naked mice, nude mice. So that's called hairless mice. And these mice actually, they don't have T cell, functional T lymphocytes. and that's when they become this hairless. And because they don't have T cells, they're highly sensitive to any kind of infection, right? So you have to put them in a germ-free environment to make sure there is no microbe in that environment. They can die. What about this animal? What is that on the back? Can you see isn't it obvious what is growing in the back of this mouse? Ear. So we use mouse to grow some of these organs or tissues. Okay, it's genetically. Sometimes what happens, the soldier in the field, right? Sometimes they lost their ear, or sometimes somebody lost their ear outer part due to fire or some kind of accident, right? And it kind of, you need some kind of ear, right? Without that, it looks bad, right? So how do you replace that? And so a lot of times what you do actually, this is actually a scaffold, some kind of synthetic scaffold, and then you place that on the back of the mice, and then it grows the tissues around it. You can remove the scaffold afterwards. And so you are growing organs using the mice. Okay, so same way, the heart valve and a lot of those parts are nowadays cultured or grown instead of, taking it from other animals. So these are all done. These are part of the tissue culture engineering. So these are pretty fascinating. What are the limitations? Good thing I mentioned, right? These animals small, take less housing, easy to handle, and you can get statistically large numbers you can use it. These animals reproduce much faster, so within 21 days you can have another litter. So we maintain our own animals and we grow them. But there are also limitations, right? The primary limitation would be these pathogens you are going to study that may not show symptoms in mice, because it's a human disease, right? The humans and mice are different, and so they may not cause disease. The most important, of course, you need the approval. This is very important. You need to have a housing, veterinary care. Caregivers must pay attention, because otherwise these animals can transmit disease, can bite, can scratch. Personal safety, as mentioned, they can attack, scratch, bite during handling. Okay, so that's you need training. Differences physiology. Human animals are different, right? Another thing is that they eat their own feces. You know that animals eat their own feces, and so it's called coprophagy. So if you are trying to study enteric pathogen or intestinal pathogen, and they are introduced certain number, and then they secreted or excreted in the feces, and these animals are going to come back and eat that feces, so it's reinfecting. So the data you will be finding is not a true infection, because they are getting more organisms by eating their own feces. Coprophagy, that's called coprophagy, eating their own feces. And so those type of problems are there. But in order to avoid that, what do you have to do? You have to put a screen, right? So that they're hanging on the screen, the feces drops below so that they cannot access it. So a lot of different ways you can overcome it. But nonetheless, there are a lot of differences. Resident microflora population diversity in rodents are different from humans. Most importantly, some animals are not sensitive to pathogens, thus may not show any symptoms. So you want to use animals for studying salmonella, for example, right? But mice are resistant to salmonella infection. But the mice are the best model to use salmonella pathogenesis, because it is easy to use, right? What do you need to do to make it sensitive? What would you do to make these animals sensitive to pathogens which they are not responding? So one would be, you saw that antibiotic treatment. You can wipe out their natural microflora. That way they become sensitive. That's one way you can do it. I have that next slide. What else you can do? You can genetically modify it, right? You saw the T cell deficient. Also you can irradiate. Radiation also wipe out your immune system. And so you can make number of different ways you could use it. And matter of fact, all those techniques and tools are available to make these animals work for your experiment. And so for this, sometimes we use different route of administration can help you. If your oral is not making the animal sick, you can introduce intravenous, intramuscular, subcutaneous, all those different routes. Neonatal animals, their immune system is weak. You can use that. they would be sensitive. And germ-free animals, you can use it, right? And also you can make them the transgenic, okay? You can genetically alter that. Also you can use immune compromised or immune deficient animals, okay? And already you saw the nude mice, athymic mice. So a lot of this technology is available and you can use them to work for you. So that's why there are all this called skid mice. You can remove T cells, B cells, and you can make a RAG deficient. We got some of those knockout animals in our system. Already I mentioned that immune compromise, right? And you can also use the suckling mice. Especially for the diarrheal diseases, they use a lot of the suckling mice, okay? And that's how we do it. And you can see this litter, anywhere from up to 9, 10 fetus you get. And so these animals are widely used for studying different diseases, okay? I'm kind of giving you a little bit more overview. So if you don't want to use the animals, can we use organs, right? Organs or tissues for research. The whole animal, now we are going to just the specific organs or tissues. And so this one again, I just mentioning a few minutes ago, can you culture organs? Sometimes we are growing heart valve in a test tube, okay? Those type of flasks they use it. And a lot of times also implantable chips they are using it. And so a lot of things are happening in the area of tissue culture. Most importantly, in our case, if we are trying to study toxins or diarrheal diseases, can we use called ligated ileal loop assay. So what it is actually, you take the animal, you anesthetize the animal, don't kill it, right? You take the GI tract out and you create a loop, okay? And then in certain loop, you'll introduce either the bacteria or the toxin. And those diarrheal toxin, their job is to accumulate fluid, right? We see diarrhea means you are losing a lot of fluid, diarrhea conditions. And then you check for this accumulation of fluid in the GI tract or the lumen. And you do that about 24 hours after you do that. So first of all, you take the animal, anesthetize it, surgically remove the GI tract, make loop, introduce the drug or the microbes or the toxin you want to test, and then you put it back, suture it, leave the animal healthy for 24 hours. You can sacrifice the animal, take the GI tract out, and look at this individual loop for fluid accumulation. Have you seen the picture? Okay. So this is how it look like. Some of them. See this loop. These are introduced with the different Vibrio population. So you can have a multiple loops though in that one animal. So instead of using 10 animals, you can use one animal and make a different section, and you can have a 10 molecules tested. These are much more data for this Clostridium E. coli heat toxin. I think this one Vibrio toxin, right? This is E. coli, and then the the different heat stable, heat label E. coli toxin. You can study this. You can see this one accumulating a lot of fluid with the high concentration of this toxin, and then the PBS low, and then the other toxins not showing that much. This one is much prominent. You can see this one. This is Clostridium perfringens enterotoxin assay, and you can clearly see all of this different. This is positive, right? You look at this. This is strong, and some others are very low, and the color is changed. Okay, so this ligated ileal loop assay can help you to study this. You can do statistics, you can do a lot of stuff, but it's coming from only one animal. You don't have to use multiple animals. Got it? This is how the loop model works. So loop is the one model. We supposed to end at 12:30, right? Just exceeded a little bit, right? Because you started late. Let me finish part of it. I don't want to restart again. So that was like a ligated ileal loop model. But you know that we also use egg, right? Embryonated egg. Egg has to be fertilized, and fertilized egg has embryo inside, right? And so usually 12 to 14 days, that's when it become. What is the total full incubation time for chicken to hatch? For chicken hen. So if you start today, starting hatching, how long does it take to give the chick? How many days? Oh, I thought all of you should know this. You guys don't grow your chicken, hen, or egg? No? You buy it? Oh, well, I shouldn't ask this question because you are vegetarian, right? That's the wrong place to ask this question. Sorry. Yeah, you should know that, right? Huh? Yeah, I know, that's to eat, right? Unfertilized egg. But if it's fertilized egg for chicken, jeez, 45 days? Okay, that's too long. How about the zoology students? Come on. You have seen the hatched eggs or the hen or the chick, right? You guys should be knowing. Come on. Huh? I hope you didn't Google it, did you? Okay, thank you. You need to be honest. I know you can Google it, you can find the answer right away. Thank you. How about the duck egg? Duck. How long did it take for duck? No Googling. Come on, these are the easy questions. Okay, so chicken take 21 days, about 21, 22 days. Duck usually 28 days, longer. Okay, depending on what animal. But the reason you need to know is that somewhere in the middle you need to have this embryo form, right? So usually about 12 to 14 days of incubation, you see this nice embryo, and you need to know also the anatomy of the egg, okay? Because the amniotic fluid, this is the sac, and many ways actually this structure looks like human system also. This amniotic fluid, how the embryo grow inside the human, many ways actually, lot of similarities. And so all of these chambers you need to know, and what you do actually, if you test the organism you want to test, right, and you introduce it or inject it, and you can crack open the cell, and then there is this amniotic, the membrane there is a thin membrane. I'm sure you all know that, right? When you crack egg, you know that there is a egg shell and the layer, and then you inject it, and once you inject it, then you close it with the tape or something, so it remains sterile. And what you are looking for, if it is a pathogen, is going to kill this embryo, and the color changes, and there is no movement, and you can check against the light called candlelight. Okay, it's very easy to do it, and that way you know within at least 72 hours you are looking for cytopathic effect, death of the fetus. In this case, and this is how you kind of figure out if this organism works. And so for different diseases, we use this egg. It's a powerful tool. Again, very simple, right? You buy the fertilized egg, bring to the lab, and you all have the incubator, right? The same incubator, you need the same temperature, adjust the temperature they will have. You can do it very easy if you are deciding or trying to use this tool. For virus culture, this is the main. All the vaccine you are getting, all done in egg. One of the questions they would ask, are you allergic to egg before you take this vaccine? Because all these viruses are grown in egg with certain exceptions. The cell culture model they use for culturing viruses, but that becomes very expensive, okay? Unless you are allergic to egg, usually those people will receive those type of alternative vaccines. But usually everybody receives vaccine made with the egg. Clear? And so egg is becoming an important tool in viral diseases or viral vaccines. So you heard about avian flu? How many of you heard about avian flu virus? It should be 100%. All of you should raise your hand, right? You haven't heard about it, bird flu? Oh, bird flu, you know. You didn't know the avian flu. Oh my goodness, sorry. It's a bird flu, right? Yes, it's a bird flu. We call it avian flu. A little bit sophisticated English, right? It's a bird flu, yes. So all of you should know, right? Okay, now ask this question. Bird flu infects bird, right? Right? Yes? That's where it came from. There's a chance it's going to come to human, right? There's a mechanism behind it. I know the answer, but I'm going to talk about that. So, if bird flu get infected, birds are dead, how we are going to protect humans against bird flu infection? That will be the worst nightmare, right? We need egg to make virus culture, right? And once we have this virus grown inside the egg, we take it out, we inactivate them or do something so that they don't cause infection, but it can produce antibody or immunity, right? And so the egg is the primary source of these viruses. Now, if the bird flu infect birds, those chicken is supposed to lay hens, supposed to lay eggs. If the birds are dead, who is going to make the vaccine? We haven't thought about it. We should be thinking about it. And that would be the nightmare. Everybody would be in big trouble. See this one? So in order to protect us, we better protect the birds first. And the pandemic, if it happens, we will be in a real big trouble. Because the birds are dying, there is no more laying hens, there is no fertilized eggs, and how we are going to make the vaccine? Large quantity in an industrial scale, we make this. Cell culture cannot support it because it's going to be very expensive. And purification of viruses from cell culture also very expensive. Anyway, I want to just give you an idea that this is, you can see a lot of those viruses, herpes virus, sarcoma virus, influenza, flu virus, mumps, herpes simplex, all of these avian flu virus, adenovirus, Newcastle disease virus, all of those are actually cultured in these different parts, and we grow them and we harvest it. Virus culture, this is essential. Okay, we use that. And next is the cultured cells. We grow the cells and we can do a lot of those experiments. I know I want to. It's already exceeded a little time. We'll catch up my next lecture. That will be tomorrow. I will finish. This is just a little bit left. It should be very interesting because these are all powerful tools. Okay, and I wish you were in my lab. I could show you all of these things we do in the lab, cell culture. These are all. This is actually a picture from my lab, and we got a lot of the cell culture experiments. Anyway, I will end it here. Any questions? Go ahead.
Sir, actually, regarding that organ culture, how is it actually the preparation?
Oh, organ culture. So normally what they do, actually they will make a scaffold. Do you understand what is scaffold? Scaffold means, is there something look like the structure, for example the ear, right? And it is made of cartilage. And you can create a synthetic cartilage type ear like structure, right? And then you add cells. You have either human cells, usually you start with the human cells, and or the secondary cells or primary cells, and then you kind of seed the cell on that scaffold. And then the purpose of these cells are try to multiply, and they will create the whole structure. They will grow. And what you do, actually, you put them in this class with all the nutrient it needs. Like in your body, you need hormone, you need protein, you need nutrients, you need amino acids, everything. It's a complex food system you have to provide. And once they are under that conditions, you put them with the under specialized incubator. I'll show you that next lecture. They will be grown in that environment. but you will be continuously feeding them, and then they will grow, and eventually they will build that specific organ. For example, heart, right? Heart valve or heart muscle. If you isolate a heart cell and you grow them in the vessel, and they will be pumping. Their job is to squeeze, right? And you will be amazed that they are growing continuously, non-stop. They are on the fast. They are just pumping continuously. It's so fascinating to watch. They will be doing that, and they will be multiplying same way. And so we create that tissue design, and then the cells are continue to multiply. But you have to seed it. You have to add cells to it. Specific cell type. So a lot of like cartilage cells, you need fibrous-like cells, you need epithelial cells. So they will design what are the type of cell populations you have. You have to add all of them, and they will grow. Okay.
So it doesn't need any host.
Any need what?
Host to culture.
Oh, host, no. They will be. So I showed you organ culture. If you want to put it in the mouse, that will be great. You know, if you have those type of system, you can do that, or you can grow them in the chamber, in a specialized chamber. I showed you some of them. That area is extremely expanding and very important, because a lot of times we try to get from other animals, like a heart valve from pig, because pig and human has a lot of similarities. They try to harvest from pig also to use that, to replace it. But sometimes we try to grow because of the fear of rejection by our body. So we have to use the human cell type to make it so that it can be implanted in humans without being rejected by our immune system. That makes sense? So that's why you have to use human cells. So a lot of cells are actually available from human, or they could start from the person maybe dead or died recently. They can harvest cells from there too, or a biopsy sample they can get. They can use that also. Okay.
Thank you, sir.
Any other questions? I know you are ready for your lunch. Go ahead, have a good lunch, enjoy your lunch, and then this afternoon.